Okay, guys, this lecture number 35 is about the cholesterol and the steroid metabolism. So the learning objectives for this one is the main lecture is the whole lecture is basically about the cholesterol. So we will look how to synthesize the cholesterol and regulation of this process, that is cholesterol synthesis. What different products can we get from this steroid and the bile acid synthesis, which are the related products, and the functioning of a special enzyme system, cytochrome P450, and the derivatives, that is the different other biomolecules that we can get from the cholesterol. So starting off with the structure of cholesterol, and it's got 27 carbons in it. Here is the diagram of the cholesterol. Important diagram can be there in the exam, and you simply have to tell which molecule it is. This is the structure of the cholesterol. So total of 27 carbons, as we can see in the diagram. There are four fused rings, A, B, C, and D in this molecule. Three are 6 carbon rings, and one D is the 5 carbon ring. So four fused rings are present. There is a peripheral side chain present, means side chain on the side, which is here attached at carbon number 17. So this one that I just highlight is the side chain, and only one double bond is present, which is between carbon 5 and carbon 6. So here is the double bond. An additional thing that you can remember is a hydroxyl group that is attached with carbon number 3 right here. Some properties of this cholesterol molecule. So it's a lipid in nature, so low solubility in water, like all other lipids. Only 30% soluble in the free form, but the majority of this cholesterol inside the body is esterified through the hydroxyl group with long chain fatty acid. So most of the cholesterol that you find in the body is attached with a long chain fatty acid, and we call it esterified. And these cholesterol esters are even less soluble than the free form. So while everybody knows that cholesterol has a bad reputation, all the doctors and the healthcare practitioners always say that cholesterol is a bad molecule, but do remember that it is essential to all the animal life, including the human beings as well. It has got very important functions to play in the body. It's a precursor, means it gives rise to many important biomolecules like steroid hormones, bile salts, vitamin D. All of these molecules we get from the cholesterol. So normal amount of cholesterol are absolutely important in the body. It's only when the amount of cholesterol or the concentration is more than normal, then only this cholesterol can become bad for the body and can cause diseases. Now there are four stages of cholesterol synthesis, and this slide is pretty important. These stages you need to remember. Now do remember these are four stages, not four reactions. The total number of reactions in this cholesterol synthesis are about 25 to 30 reactions. But there are four main stages, so every stage can have more than one reaction, and you have to remember these four stages. Stage number one is we take three acetate molecules or three acetyl CoAs, as we can see in the diagram as well, join them together. Process is called condensation, and we give rise to the molecule mevalonate. So stage one, three acetates to mevalonate. Stage number two is mevalonate is converted into a 5 carbon molecule known as isoprene. So stage 2, mevalonate into activated isoprene, which is a 5 carbon molecule. As we can see here, this isoprene molecule has got 5 carbons in it, and with it they are attached to phosphates as well. Stage number 3, we take 6 of these isoprenes that we made in stage 2. We take 6 such molecules and polymerize them, join them together to make a linear structure which will have 30 carbons, because each isoprene has 5 carbons. We used up 6 isoprenes, so total carbons becomes 30, and it will be a linear structure known as a squalene. So stage 3, 6 isoprenes into squalene, and then this linear molecule will cyclize, means rings, or 6 carbons and 5 carbon rings will be formed here, as we can see these 4 rings in the diagram. This is squalene, even though they look like in the ring form, but the rings are not closed. It is a linear structure. So stage 4 squalene cyclizes into the cholesterol molecule, and there will be 4 rings, as we saw in the first slide as well, in this cholesterol molecule. So next up are these 4 stages or 4 steps in a little bit of detail. Step number 1 is important, guys, because it has the rate limiting reaction in it. Otherwise, you don't really have to go into the details of reactions in each of these remaining steps. But step number 1 is important, especially the rate limiting step, which is here. So I will quickly go through it. The main step is the formation of this molecule mevalonate from the acetyl-CoAs. We will use 3 acetyl-CoAs in total. So in the first reaction, we take 2 acetyl-CoAs joined together to give us a molecule aceto-acetyl-CoA. Then we add up one more acetyl-CoA in it, and this becomes HMG-CoA. Remember the short name, which is very commonly used. Full name is beta-hydroxy beta-methyl butyryl-CoA. So you can simply call it HMG-CoA. So step 1, 2 acetyl-CoAs into aceto-acetyl-CoA. Step number 2, aceto-acetyl-CoA plus one more acetyl-CoA, which is here, to give us HMG-CoA. And the last step, or the last reaction of this step, is the rate limiting step, which is HMG-CoA into mevalonate, done by HMG-CoA reductase. Very important enzyme in the body, guys, one of the most important enzymes, and this catalyzes the rate limiting step and the point of regulation. So if you want to control the whole pathway, we will control this step or this enzyme HMG-CoA reductase. 2 NADPH molecules are used in this reaction. So in order to synthesize cholesterol, we need these NADPH molecules. Then we have step number 2. As I said earlier, you don't need to memorize all of these reactions in step 2, 3, and 4. But step 1, the rate limiting step, is important. So the whole step is going to be conversion of the mevalonate that we made in the first step, converting it into 2 activated isoprenes, each 5 carbon long. So mevalonate is first converted into 5-phosphomevalonate. We simply add up a phosphate group to it, so mevalonate becomes phosphomevalonate. ATP is used in this one. Phosphotransferase is the enzyme. Then we attach a second phosphate to it. The first one was already there. Then we attach the second one, and it becomes pyrophosphomevalonate. Kinase is the enzyme this time. And finally, the third phosphate is attached to this molecule with the help of decarboxylase. So in first 3 steps, we have simply added up 3 phosphates one by one. These were the already attached ones, these 2 on the right side, and the recently, or the third one, is right here. Next step is decarboxylation, in which we remove a phosphate and also carbon dioxide from it, and it is going to be converted into the activated isoprenes. So these isoprenes have got different names. This one is the first one formed is isopentenyl pyrophosphate. The short name for this one, if you want to remember, you can simply remember it as IPP. So isopentenyl pyrophosphate, or IPP, is the first of the activated isoprenes. And the second one made is dimethylyl pyrophosphate, or you can simply call it TMPP, dimethylyl pyrophosphate, or DMPP is the name of the second isoprenes. So we made the isoprenes in the last step. In this step, we take 6 of the activated isoprene units, join them together, and make a 30 carbon squalene molecule. Uh, all of these activated isoprenes are 5 carbon molecules to remember. So when we take 6 of them, join them together, 6 multiplied with 5 will give us a 30 carbon squalene at the end of it. So this whole step is simply adding up or joining up the isoprenes together. So we take the two that we made in the last step, IPP and DMPP, and make a geranyl pyrophosphate, which is GPP. So IPP plus this DMPP in the first step join to give us a GPP, which will have 10 carbons in it, because each of these two had 5 carbons in them. So 5 plus 5 carbons gives us the GPP, which has 10 carbon molecules in it. Then we take another one of this IPP and join it with GPP. So GPP plus one more IPP gives us this farnesyl pyrophosphate, or FPP, which has got 15 carbons, as 10 plus 5 of this isopentenyl pyrophosphate gives us the 15 carbon molecule, which is known as FPP, or farnesyl pyrophosphate. And in the last step, we are simply going to add up one more FPP. So one FPP 15 carbon plus one more FPP 15 carbon, joining these two together gives us the 30 carbon squalene molecule, which is a linear structure. Do remember, it does not have the closed rings in it. Next up is just cyclization, conversion of this squalene into the steroid nucleus, or the cholesterol molecule, which will have 4 closed rings present in it. So squalene is first converted into squalene epoxide with the help of a squalene monooxygenase enzyme. And then we have multiple steps with the same enzyme cyclase that converts it into lanosterol, and finally lanosterol into the cholesterol, which has the same structure as we saw in the first slide. 4 fused rings, 1 arch chain, and 1 hydroxyl group plus 1 double bond between carbon 5 and carbon 6. Next up is the fate of these cholesterol that we synthesized. What will happen to this cholesterol? How will the body use it, basically? So invertebrates like us human beings as well, most of the cholesterol is synthesized in the liver. So do remember, the main organ of synthesis of cholesterol is the liver, and then exported means sent out of the liver to be used by other organs. And the form in which it is exported, it can be bile salts, bile acid, biliary cholesterol, and cholesterol ester. So these are 2 or 3 different molecules. In this form, cholesterol is exported out of the liver. Some of the cholesterol is also used inside the liver, and the main purpose is for membrane synthesis. So its use in the liver is for the synthesis of some membranes. As this cholesterol, do remember guys, one important function is it takes part in the synthesis of membrane of the cells and of the organelles inside the cell. Now once this cholesterol is exported, it will go to different tissues, and in other tissues this cholesterol can be used to make steroid hormones, which are plenty of important hormones: testosterone, progesterone, estrogens, aldosterone, and so on. So all of these hormones can be synthesized by cholesterol, and they have an important role. And it can also be used to make a vitamin D inside the body as well. So next up is the fates of cholesterol in a diagrammatic view. More or less the same information that was there on in the previous slide. So here we have the cholesterol mostly synthesized in the liver, and it can be converted into steroid hormones after being exported out of the liver. So we have got special organs in which these steroid hormones will be synthesized. Other hormones like hydroxysterols can be formed from cholesterol as well, and we can attach a fatty acid with this cholesterol, convert it into the cholesterol ester, and sometimes convert the cholesterol into bile salts as well. One example of bile salt is given taurocolic acid, and these bile acids help us in lipid digestion. So this slide has got the conversion of cholesterol into the cholesterol ester. Again, very important reaction and very common reaction in the body, where we attach a fatty acid, the activated fatty acid known as fatty acyl CoA. We attach it with the cholesterol, and the product is known as cholesterol ester. Here we have the fatty acid that is attached at carbon number 3, where initially the hydroxyl group was present. ACAT is the name of the enzyme. As I said, very important enzyme and very important reaction in the body. ACAT stands for acyl-CoA cholesterol acyl transferase. So the cholesterol esters are more nonpolar than the cholesterols. They contain a fatty acid joined or esterified to the oxygen molecule, as we can see in the diagram as well. Fatty acyl CoA is the activated fatty acid basically that will provide the fatty acid, and these cholesterol esters they are transported in lipoproteins to other tissues, or they can be stored in the liver. So this point is important. For transportation, these cholesterol esters are put in these molecules known as lipoproteins. Lipoproteins are the transporters for cholesterol esters, and these lipoproteins detail will come up later on. They are basically packaging molecules. The surface of these lipoproteins are made up of special protein known as apolipoprotein and a bit of phospholipid. So outside it's protein and phospholipid, and in the interior of these lipoproteins we have got cholesterol, DHEs, and the cholesterol esters. Important thing to notice, this ACAT enzyme is activated by the cholesterol molecule itself, which is the substrate. So cholesterol or substrate is activating the ACAT. Next up is regulation of this whole process, which is done by the rate limiting enzyme HMG-CoA reductase, which was present in the first step of cholesterol synthesis. And this enzyme is controlled by the covalent modification mechanism, which will provide the short-term regulation. So we can change this modification in very short time. The most important thing to remember out of the slide is this HMG-CoA reductase will be active when it is dephosphorylated. So dephosphorylated form is the active form. When it becomes phosphorylated, it becomes inactive. And the phosphorylation and dephosphorylation simply means attaching of the phosphate group or removal of the phosphate group, as we can see on the right side here. So without the phosphate is the active form, which we also call dephosphorylated form. With phosphate, the P that is attached here or with this left side enzyme is the phosphate. So with phosphate is phosphorylated form, and it is the inactive form. Now there are different things that can activate it or inactivate it. An important enzyme is this AMPK. So AMPK enzyme phosphorylates and therefore inactivates, or you can say inhibit the HMG-CoA reductase. So this enzyme AMPK, AMP dependent protein kinase, shortened as AMPK, this will inhibit HMG-CoA reductase. We have got two hormones, glucagon and epinephrine. Both of these causes the phosphorylation of this enzyme, and therefore decrease its activity. In other words, we can say inhibits it or makes it inactive. All mean the same thing. Whereas the only molecule that is going to activate this enzyme is insulin, and it will do so by dephosphorylation. It is simply going to remove the phosphate from it, dephosphorylate it, and therefore increases the activity, as we can see here. So two groups of molecules inactivated AMPK enzyme and the glucagon and epinephrine hormone, whereas insulin is the activator. Next is a group of drugs that are very commonly used in the medical practice these days, known as the statin drugs. And the main function of these statin drugs is to lower the cholesterol inside the body. As we know that high cholesterol levels can be dangerous and can cause a lot of diseases in the body. Therefore, these statin drugs are used for the patients who have high cholesterol. And the main mechanism by which these drugs act is they inhibit or they stop the rate limiting step, which we just saw in the previous slide, HMG-CoA reductase. So this is the enzyme that controls the whole pathway. If we can inhibit or stop this enzyme, we will simply stop the synthesis of cholesterol. So we give the statin drugs and they go inside the body and stop this enzyme. Therefore, cholesterol synthesis is stopped and cholesterol levels are decreased inside the body. The statin drugs resemble the mevalonate, so they have similar structure to the mevalonate, which is the substrate of this enzyme. So the mechanism of action is competitive inhibition. So these statin drugs competitively inhibit the HMG-CoA reductase enzyme. Again, an important point to remember for the exam point of view. The mechanism by which these statin drugs inhibit the HMG-CoA reductase is competitive inhibition. So they lower serum cholesterol, as we just saw. There are some side effects of these drugs as well. Muscle weakness is seen, coenzyme Q depletion, which is a part of the electron transport chain. But they are going to improve the circulation, the blood circulation in the body, stabilize the plaques by removing cholesterol. So remove cholesterol from the plaques and therefore reduce the vascular inflammation. Basically help in the blood flow process and save the patient from the cardiovascular diseases. Some examples of these drugs, the statin drugs are compactin, simvastatin, pravastatin, and lovastatin. So most of these names end at the statin. That is why the group is called statin drugs. So next is regulation of the cholesterol metabolism. The most important, we already saw HMG-CoA reductase enzyme, the rate limiting step, AMPK, and there are two hormones, so AMPK and the glucagon hormone, plus the epinephrine as well, not written here, you can add it up. They have the negative effect on this HMG-CoA reductase, therefore it will stop the enzyme, stop the synthesis of cholesterol. Low levels of ATP also has a negative effect, as we can see the cross here. Plus, if there is too much cholesterol in the body, it can be converted into this molecule oxysterol, and this will loop back by negative feedback mechanism and stop the enzyme as well. Plus, it also stops this LDL cholesterol conversion to cholesterol. Whereas the only thing that has got a positive effect we already saw is the insulin. So insulin, when it is in high amount, it will always activate the HMG-CoA reductase enzyme and therefore increase the synthesis of cholesterol. So this slide is mainly about the molecules that we can make from the cholesterol, the derivatives of cholesterol. To be specific, the hormones or the steroid hormones that we can make from the cholesterol molecule. So there are several classes of this cholesterol-derived steroids. Most of these are hormones. There are two main glands, adrenal glands and the gonads that will synthesize the steroids. First up, cholesterol is converted into pregnenolone, which is a precursor molecule. Then we have the first of the hormones, progesterone, and from this progesterone we synthesize all the other different hormones. So in the adrenal gland, we can make two types of hormones, mineralocorticoids and glucocorticoids. An example of mineralocorticoid is given, known as cortisone, and later on this cortisone can be converted into another one, aldosterone, as well, which regulates the reabsorption of sodium and chlorine in the kidneys. This progesterone can be converted into a glucocorticoid as well, which example is this cortisol hormone, again an important hormone that affects the protein and the carbohydrate metabolism mainly, or in other scenarios, in the gonads especially, this progesterone will be converted into the male sex hormone testosterone, which later on can be converted into estradiol or estrogen, which is the female sex hormones. So these are male and female sex hormones, influences the secondary sexual correctors and regulate the female reproductive cycle as well. So a similar diagram, more or less the same information, but one important thing is added up in this one, which is the conversion of cholesterol to pregnenolone, which is done by this enzyme desmolase. So this enzyme is very important, which converts cholesterol to pregnenolone. Later on in the ER or endoplasmic reticulum, we get the progesterone, and as we saw in the previous slide, progesterone can be converted to testosterone, the male sex hormone, later on converted to female sex hormone estradiol, or we can convert progesterone to cortisol, and in other organs into aldosterone as well. So here are the first two steps in a little bit of detail, from cholesterol to the pregnenolone, which later will be converted into the steroid hormones. So two important enzymes are here, mixed function oxidase that converts the cholesterol into dihydroxycholesterol, and then the desmolase, which is the more important of the two, converts dihydroxycholesterol into pregnenolone, and this is the rate limiting step of this whole pathway of converting cholesterol into the steroid hormones. And pregnenolone, to remember, is the precursor of all the steroid hormones. So during the synthesis of cholesterol, we saw this molecule isopentenyl pyrophosphate, which was an activated isoprene, and I told you that IPP is the short form. So these activated isoprenes are very important molecules in the body and have many alternative fates, or you can say very different functions to play for. One function we just saw in this slide, in this lecture, sorry, that it can be converted to cholesterol, and later cholesterol can be converted to vitamin D, bile acids, and the steroid hormones as well. Apart from cholesterol, this IPP can be used to make different vitamins in the body, vitamin A, E, vitamin K, different carotenoids, and some plant hormones as well, not in the humans. And it can be used to make rubber, phyton chain of chlorophyll, which is the main pigment forming molecule in the plants. Dolichols, and quinone is an important one regarding the humans, which is an electron transport carrier. So do remember this quinone, which is a part of the electron transport chain of the of the body. So IPP can be used to give us this molecule, and other isoprenes as well can be converted from this one.